Okay, let's get started. Yeah. Okay, so welcome to week 7 of Computational Systems Biology. My name is Shomo. I am PMRFTA from IIC Bangalore, and this course is taught by Professor Karthik Raman of IIT Madras. Yeah, so last week we discussed about evolutionary algorithms, genetic algorithms, how you can use the concept of evolution to create algorithms that can solve for different equations and parameters. Today we will look into specifics of a type of analysis of systems biology, which is uh, flux balance analysis. I hope I'm audible. Yeah. Okay. Okay. So firstly, before we move into the particular analysis part, we need to define a few metrics and a few objects, basically. So this is what is called a stoichiometric matrix. So I hope you're familiar with the stoichiometric matrix because this is quite common. So what it is, it's basically if you write an equation ax plus bx giving c, ax plus by giving cz. So your a, b, and c are your stoichiometric coefficients of your equation of your reaction. So these elements in a matrix form is basically the stoichiometric matrix. So for example, if you have a reaction which is given by this form, a plus b equal to 2c, and the uh, stoichiometric matrix, or the stoichiometry of the individual elements of this equation. For a, it's -1. So keep in mind of the direction of the equation as well. So a is getting used up. So the coefficient will be negative for a, and the coefficient will be negative for b. But since c is getting formed, the coefficient of c is going to be positive. So it's like -1, -1, and +2 for a, b, and c respectively. Now, like, this is what a stoichiometric coefficients mean. So if you assume a metabolic network which has a set of these reactions, so this was one reaction. Since if you consider a big metabolic network, you have a set of reactions inside the network which are like uh, like creating new molecules, new products, which are getting used up by other reactions in the network as well. Now, if you assume that one such reaction is Ri, I to I in the subscript, so one of these R1 to RR. So Ri is one such equation, and you assume that the rate is Vi given by V under subscript I. Now, if you collect the individual reaction rates of each of these equations and you turn it to a vector, so basically v1 comma v2 comma v3 up to vr, and if you turn it to a vector, it's basically going to be a transpose of this whole row matrix. So you convert it to a column matrix, which is the vector. Now, if this is your rate vector, similarly your concentration vector will be given by each of the elements inside in the reactions, and sorry, and it actually varies with time. So your concentration is going to be a function of time, because uh, as time passes, the concentration will change depending on how the reaction is going. So this is also a similar situation. You take all the concentrations of each of the molecules in the reactions, in all the reactions, and you do a transpose of that. So that gives you the concentration vector. Now, what is the stoichiometric matrix? The stoichiometric coefficients of a reactions are collected in the vector SR. So this is this vector. No, actually, this the stoichiometric vector is not defined here. This is actually the stoichiometric vector. It's the same thing. So you have like all the elements of the reaction and their corresponding coefficients, reaction coefficients. So those bring up the stoichiometric coefficient vector, and this is given by SR. So in SR, there is one position for each metabolite in the metabolic system, and the coefficient, stoichiometric coefficient of the reaction are inserted to appropriate the position. So if you have something like a plus b giving 2c, For A it's going to be -1, for B it's going to be -1, for C it's going to be 2, right? And the zeros are there for other elements in the reaction. So basically, if you are considering this particular reaction, there might be in the whole network, there might be other metabolites also involved in the in the whole network, but you are, for this particular reaction, you are not considering the other metabolites, so rest of them will be zero. Only the elements inside that are involved in this particular reaction will be given a coefficient in the vector. So that is what it is done here for this particular reaction, A plus B giving 2C. And if you combine all these particular individual coefficient vectors into a big matrix, it is what gives you the stoichiometric matrix. So in the matrix S, this there is one row for each metabolite M1, and obviously dots if it is not present in that particular reaction, and till MM. So it's situation similar situation like this. So each of these row, so if you consider this as a matrix, so A the coefficients of A are given by this particular row if you like expand it. So for example, if you assume this particular row is for A, so all the coefficients are given by this particular row only for all the reactions A is involved in. So basically what it is doing, all your columns are your individual reactions and your rows are your individual metabolites. Now all the metabolites will not be involved in all the reactions, so in some of these vectors you'll find a lot of zeros and only a few set of non-zero coefficients which will denote that only those metabolites are involved in the reaction. The coefficients Sj along the jth column are the stoichiometric coefficient of the reaction J, as I was saying. So each of the column denotes a particular reaction, and each of the row will denote a particular metabolite. I hope I'm clear till now. Now the question comes, when will this system or when will this metabolic network will reach steady state? When after what time, or like at what concentrations of particular elements inside the network, which are metabolites, does this particular network go into steady state? So finding steady state from a mathematical perspective, it's not that difficult, but if you have big matrices, big equations, it might be difficult algebraically because you need to solve a lot of equations, but technically the idea is same. Basically, if you consider the idea is that if you differentiate your equation at that particular point of time at steady state, the dx dt, or the rate of change of concentration of the metabolites inside the network, will be 0. So that is the idea. So this is the final equation for this. So dxi dt, xi denotes each of these individual metabolites, and dxi dt, if you differentiate it with time, it's basically giving you 0. Now where does this SV come from? So that is something that we'll discuss here. So if you consider dx dt, basically all the system's equation of different equations together can be expressed as a matrix form. So your S is your basically your velocity vector, V is your velocity vector, and S is your reaction vector. As a product of those, that can be equated as the rate of change of concentration of a particular metabolite. So S is your reaction vector, V is your velocity vector. You multiply both of them, that is actually what is given by dx dt. Now like this individual dx x, when you're considering, that is one single metabolite. Now there will be like n number of these metabolites, so you need to consider all of them. So what dx dt becomes is not a single entity, it also becomes a vector. So it also becomes a vector, each of the element denoting dx1 dt, dx2 dt, dx3 dt up till dxn dt. So if you imagine, on the left hand side you have a vector of these differentials of your metabolites, dx1 dt, dx2 dt, dx3 dt, etc. etc. On the right hand side you have the vector product, the matrix product of your set of your reaction vector of each of these of these equations. So for x, you have one reaction vector. For x2, you have a reaction vector. For x3, you have another. So you what you are doing is, on the left you have the vector of all your metabolites, differential of those metabolites with respect to time. On the right hand side, you have the whole multiplication of your reaction vector and your velocity of the reaction vector, both of them. So the thing is, if you want to find certain solutions of these equations, so for example, why are we interested in steady state? Because you assume that after a certain period of time, a substantial long period of time, the system will be in equilibrium, because that is how a system works. Because after a certain period of time, you assume that the system will be in equilibrium. So that is where the steady state concept comes into. So what you're doing is, basically, you're finding concentrations of these particular metabolites at this steady state, where your dx dt equal to 0. So basically that is what is denoted here. So the concentration of metabolites remain constant over time. Thus the derivative of your concentration is 0, which is the definition of steady state. So this equation gives you a steady state. So I to j is basically I is the number of, I is denoting dxy, xi, x1, x2, x3, x4, etc. till xi. So x and x to the subscript I is giving you all the metabolites inside your model or inside your network, and j equal to 1 to r is giving you all the reactions inside the particular network. So this is an equation that is used to find the steady state of a particular metabolic network, and it can be other type of networks as well, but the idea is same. The core concept is that your concentration of your metabolites or your reaction compounds, they should not change with time. So that is the general idea. Okay. Now let's talk a little bit about how the dynamics inside this metabolic networks work. So there's the whole network. Inside this, the reactions are going on, but it's not a static network, right? Because some things are getting formed, that thing is getting used up by some other reaction, it is forming something else, that is getting used up by some other. So there's always a dynamic inside this kind of reaction networks, specifically metabolic networks. So so firstly, let's talk about the representation. As we talked about, metabolic reactions are depicted using the stoichiometric size m, s sized m cross n, and in each row s, it denotes a unique compound, which is the metabolite. So as you remember, each of these the row information will give you metabolite information, and the column information is going to be your reaction information. Entries in the column denote stoichiometric coefficient of participating metabolites, negative for consumption, positive for production, and 0 for non-participation, as we talked about in the previous slides. Due to limited involvement of metabolites in most biochemical reactions, S is predominantly sparse. That is intuitive, because you have, say for example, 50 reactions going on inside in a particular metabolic network, but you have 100 or 200 metabolites, right? So all of the metabolites will not be involved in all of the reactions. So most of the elements inside the matrix is going to be actually 0. So what it becomes is a sparse matrix, predominantly sparse matrix. So that makes it easier to handle this kind of data structures, because if it is sparse, it's going to take up less space, and it is going to be easier to do operations on. So it is actually a good thing for us. Now one new terminology that is getting introduced here is basically flux. Flux through the reactions is represented by the vector v of length n, and concentration of metabolites is given by the vector x of length m. So this m cross n is coming from here. Just a second. Okay. Now, yeah, that it's a mathematical formulation of flux. So what is it's given by the rate your velocity vector. Now, intuitively, what do you think is flux? So in a metabolic network, the flux is basically uh, the rate of metabolite flow through a pathway inside the network. So basically how much flow of metabolite is happening through the network. So if you are from engineering backgrounds, or if you remember class 12 physics, we studied something called flux of electrostatic flux. So basically how much charge is going through a particular area or a particular element. So the concept is kind of similar here, is how much metabolite is getting uh, flown through a particular pathway, or the basically what is the in vivo rate of an enzymatic reaction. So that the rate of the reaction, why we are talking about the velocity vector here, because the velocity of the reaction is somewhat intuitively connected to the flux of the network, right? So one definition that is there in Wikipedia is basically telling that the rate of turnover of molecules through a metabolic pathway. So it's the same thing. Intuitively, if you think about it, it's basically how much of the metabolite is flowing through a particular pathway or a particular reaction inside the network. When you're talking about particular reactions, if you're talking about the whole network, you can calculate the network flux of the whole network, even considering all the reactions inside the network, right? We'll talk about flux a lot, because flux balance analysis is something that is there in the syllabus, and we'll talk about that a lot. Okay, so at steady state, what are the mass balance equations? As we saw here, it's basically dx dt equal to summation of these particular product of your reaction and your velocity vectors, and equated to 0. So it's the same thing here, as v equal to 0, and any v satisfies this equation resides in the null space of S. So this is a matrix concept. If you are like not familiar with it, it's not that difficult to understand. So basically, when two vectors are having a product of 0, it means the cos theta between the theta. So if there are two vectors like this, and the angle between them forming is theta, and if you multiply, this is one vector, this is one vector. So it is in geometric form, but in your algebraic form, this is actually a column matrix. This is also actually a column matrix. Now if you multiply both of them in a geometric form, it means that the angle between them will be considered. So the dot product is AB cos theta, right? So if you A dot B, if you do a dot product, it's basically AB cos theta. Now if the angle between the two vectors is 90 degree, the cos theta becomes 0, and the product of the two vectors becomes 0. So what it means is both the vectors are at 90 degrees to each other. So this vector does not have any influence on this vector. That is what it means in a geometric form. Now, in a more nuanced version, what the definition is, if this is a vector, the angles of this vector forming 90 degree with other planes in the same dimension, it's called the null space of this particular vector. So this is a vector, all the vectors that are at 90 degree to this particular vector, this can be this, this can be this, this can be this, any of these vectors are basically, all of these vectors together are forming the null space of this particular vector. So that is what a null space means. What in geometrically it means that it is at a 90 degree angle to the original vector, and intuitively what it means is that this vector does not have any influence on all the vectors that are present in the null space of this particular vector. So that is what it means. So if you like do not understand the terminology at any point of time, please let me know even. Okay. So yeah, that is the steady state equation for mass balance, right? So SV equal to 0. Now, if you try all these techniques in real world metabolic networks, there are some issues that you're going to face. Firstly, they're going to have more reactions than compounds, right? N will always be greater than M. oh sorry, I think I gave an example a few minutes back where I talked about the the reactions uh, being lesser than the number of compounds. So it's actually the other way around. So the compounds in real life networks, what will happen is the reactions will be much greater than the compounds. But these compounds, one compound will be involved in like 5-7 of these reactions, but not all the compounds will be involved in all the reactions. So that is why you have a lot of zeros, a sparse matrix there. So yeah, just to clarify. This leads to an undetermined system where the number of unknowns exceeds equations, hence lacking a solution. So what happens is, if you model all these reactions, so the number of unknowns, the number of the differential number of equations that will be formed will be proportional to the number of reactions that you have, right? And if you have a high number of reactions, you have a high number of equations, but the number of compounds there is much lower. So it becomes a number of what happens is the number of unknowns exceeds the number of equations, so it actually lacks a unique solution. So what we do is we satisfy certain constraints. So what we do is we put some constraints on this system and solve the equations with these constraints, which helps us get the solution. We'll see a couple of examples, I think, today only. Okay. Despite constraints defining solution ranges, pinpointing specific points within the solution space is feasible. Examples include identifying maximum growth rate or ATP production under organism-specific constraints. And flux balance analysis is actually a method for pinpointing optimal points in this constraint space. So you have a two-dimensional space, and you have a region inside that space where your solutions might lie. But you do not know, you cannot pinpoint the solution. It's bound by certain constraints, but it's pinpointing that solution is very difficult, and flux balance analysis actually comes in handy at that point of time. Okay, before we go into flux balance analysis, we were talking about constraints, putting constraints in different regions in your solution space to find the most solution that is feasible. So this is what is known as constraint-based modeling. So it is an approach based on the assumption that organisms exist in particular environments that typically have scarce resources, and over time the probability that the fit will survive is higher compared to the probability of the survival of the less fit, which is basically the evolutionary concept. To be fit for survival, a myriad of constraints must be satisfied, which limits the range of available phenotypes. So when you are considering a system, specifically a biological system, it is actually governed by a lot of constraints. There's a constraint on the amount of resources it can use, there's a constraint on the amount of energy it can use, there's a constraint of this amount of protein it has to produce, or etc. etc. So there can be a lot of constraints. Some of these are like very very obvious. Like there is obviously not unlimited resources in the system. Sorry, are you saying? Okay, sorry, I thought you were saying something. Okay, yeah. Now these constraints can be subdivided into specific broader categories. Mainly 4 I have described here. First is the physiochemical constraints, second is the topobiological constraints, third is environmental, fourth is regulated. Now physiochemical constraint is basically your basic laws of physics must be obeyed. Mass, energy and momentum must be conserved. So that's the basic laws of Newtonian mechanics, and it should obey all these laws. Furthermore, biochemical reactions must result in a negative free energy change to proceed in the forward direction. So if you remember free energy, Gibbs free energy change and everything. So if it's an exothermic reaction, if the energy of the product is lower than the energy of the reactants, then it's an exothermic reaction. So it goes like this, and it goes at an energy that is lower than that of the reactants. Now this is what is an exothermic reaction, and the Gibbs free energy change, the delta G, is actually negative. So that is what pushes a reaction in a forward direction. So if your product is at a lower energy level, then your reactions will automatically be converted, reactants will automatically be pushed forward in the forward direction to form products. So that is quite understandable. Topobiological constraints include, say for example, crowding of molecules inside the cell. So if you, it's a similar type of concept is there in chemistry, which is the static hindrance. So there must be enough space and less crowding of molecules. There should be enough space between two molecules so that certain changes of the molecules can be allowed. So if all the molecules are crammed together, adding a particular methyl group to a particular molecule will be very difficult, because there is space crunch, because there's steric hindrance. There is no way that the methyl group can go and bind physically to that particular carbon of a compound, because around it it's so crowded that it does not have the space to go and bind. So that is what steric hindrance is. It's a similar type of concept here of topobiological constraints, right? And also there is a limited space inside the cell or inside the mitochondria or wherever this particular set of reactions are taking place. There is a 3D constraint there, there is a space constraint there. And so increasing the concentration of the reactants or the products to after a certain level will actually not improve the reaction rate, because there is no space for the new compounds to be formed or new groups to be added to a particular compound. So it becomes difficult like that. Environmental constraints, which is basically nutrient availability, pH, temperature, osmolarity, availability of electron, etc., etc., etc. So as you remember, I think so every enzyme has a particular pH and a particular temperature at which it works best. And if you change the temperature slightly here or there, the pH, it will the reaction will not be an ideal reaction, because the working of the enzyme or the different reactants inside the reaction is basically a function of temperature. So there's one particular temperature at which it has the uh, highest activity, and if you move away from that left or right, the activity drops. So those are environmental constraints. Regulatory constraints differ from all this category, as these are not actually hard constraints, but more of regulatory constraints. So basically they are implemented by cells in various ways, including the amount of gene products and their activity. So the cell, using signaling and other mechanisms, actually constrained. A lot of regulation actually is going on. That is what is being explained here. So when a particular reaction is undergone, a particular reactant is getting converted to a product, the cell is actually regulating a lot of that in terms of the reactants available, the products available, the pH of the cell, the temperature of the cell. Everything is getting regulated by the cell. The cell is actually regulating these reactions in certain ways, and like there is always a feedback mechanism for the cell. So if it exceeds a certain concentration, there will be a feedback mechanism to actually lower down the reaction rate or something like that. So that is also a kind of a constraint that you need to consider while doing this kind of modeling. Okay, now how do you mathematically represent these constraints? So after the recognition and definition of these constraints, they need to be described mathematically. Once in mathematical form, they can be used to apply or perform an in silico analysis. So two things: one is bounds, one is balance. So balance is basically constraints that are associated with conserved quantities, mass, energy, these kind of things that we were talking about. And bounds are constraints that limit numerical ranges of individual variables and parameters, such as concentration, fluxes, or kinetic constraints. So what it means is you need to always follow a mass balance approach or a energy balance approach in the particular reaction or the network. So that is the kind of a balance that can be used to simplify the network. And second thing is bounds. Like this particular compound can go up to this concentration only. When it reaches this concentration, this particular reaction rate slows down, or this particular reaction gets completely cut off. So these kind of nuances that you can use to actually simplify the equations a little bit, and do your modeling. The conservation of mass is an example of a balance constraint. Mass balance is utilized in terms of flux and the stoichiometry of each reaction. And if you consider S be the stoichiometric matrix containing all the reactions in the network, then it's an M cross N matrix where M is the number of metabolites, N is the number of reactions. And the entry Sij defines a stoichiometric reactant I in reaction J. So basically, this is what they are talking about. Now, now let's talk about flux balance analysis. So it's actually a constraint-based, a balance-based approach. Yeah, sorry. Yeah. So it's a constraint-based and a balance-based approach where basically you use certain techniques to find the optimal solutions of your set of reactions or a set of equations. So this is basically a kind of a simplified workflow on how FBA works. So you have a set of metabolic reactions. You curate them from literature, or you get them from your experiment or whatever. You have a set of metabolic reactions, and you make a whole network. You formulate the stoichiometric matrix. So on the each of the row denoting a metabolite, and each of the columns denoting a reaction. Now after you form this matrix, you use mass balance constraints. So yeah, like we'll talk about this maybe later, but basically you apply constraints. Not always mass balance, it can be energy balance, it can be something else. Some constraints you apply to that, and then you define an objective function or a cost function which you need to minimize or maximize, and then you optimize Z using linear programming, LPP, if you remember. We'll talk about an example of this in the later slides. So first, a metabolic network reconstruction is built consisting of a list of stoichiometric balanced biochemical reactions. So yeah, that is one more thing that is important, is the reactions must be balanced. So when you're doing the stoichiometric matrix and forming this matrix and doing all these analysis, your equations must be balanced. You cannot use unbalanced equations. So firstly that, yeah. Next, this reconstruction is converted into mathematical model by formulating a matrix S, which is the stoichiometric matrix, in which each row represents a metabolite and each column represents a reaction. At steady state, the flux through the given reaction is given by the equation SV equal to 0. We talked about that in the earlier slides. And since there are more reactions than metabolites in large models, there is more than one possible solution to this equation, as we discussed. Now an objective function is defined where Z equal to C transpose V. Where C is a vector of weights. Weights, when you talk about weights in a network or weights in a matrix, it basically indicates how much importance does this particular element have in the context of the whole thing. It can be a network or it can be an equation, a simple one-dimensional reaction. So that is what a weight of this particular basically this is how you define the weights, right? So your C vector will be a vector of weights, and your V is your velocity vector, as you will always find out. And in practice, when only one reaction is desired for maximization or minimization. So this is the most important part. Why do we want to maximize or minimize a particular reaction? Say for example, you have taken, I think I'll give this example only. You have taken strains of E. coli, and you have used transgenic ways to insert the insulin gene in the E. coli genome, and you expect the E. coli bacteria to actually produce the insulin from the genetic information that you have provided. Now that will happen, but there is a lot of constraints involved in that. So the gene that you have transferred in, that it actually is not a native gene of the particular strain of E. coli as well, right? So it's a human gene. So the kind of product for that will be formed is not going to be exactly same to the human counterpart. Is going to be certain differences. But actually, if you know that some nascent peptides are also formed in the E. coli engineered version of insulin formation, so a certain nascent peptides are formed which needs to be cut off actually before injecting into a human. So certain modifications will be there because your E. coli is a prokaryote and we are eukaryotes, so we have post-translational modifications and these kind of things which the E. coli does not have. Now what you want to do is you want to find out a way where you can maximize the production of insulin using all the resources that you have. So that is your problem statement actually. That is your cost function. So your cost function or your objective function, you are defining in a way that you want to maximize the production of a particular substrate or a particular metabolite. That is your idea. So that is why you are maximizing or minimizing a particular cost function. Yeah. This is the objective function. Z equal to C transpose V. C is a vector of weights which indicates how much each reaction contributes to the objective function. In practice, when only one reaction is desired for maximization or minimization, C is a vector of zeros with a 1 at the position of the reaction of interest. Obviously, when simulating the growth of the objective function, we will have a 1 at the position of the biomass reaction. Right. So just to reiterate, you have a set of metabolic reactions, you know their coefficients and everything. You formulate the matrix for your stoichiometric coefficients, and then you apply mass balance constraints and other kind of constraints on your particular matrix or your model, and then you ultimately define a particular objective function that you want to minimize or maximize a particular thing, right? And finally, after all this objectification and everything, you want to find a set of solutions that actually satisfy all of the constraints and all of the objective functions that you have defined. So finally, linear programming can be used to identify a particular flux distribution that maximizes or minimizes this objective function while observing the constraint imposed by mass balance equations and reaction bounds. We will see 1 or 2 examples later in the slides. Right. So so we talked about FPA, the idea of FPA and how FPA is applied across different conditions. Now if you want to kind of use stoichiometric network reconstruction from FPA. So basically, a stoichiometric network reconstruction is assembled piece by piece with gene-protein reactions relationships assimilated from experimental data. So this is your experimental data. You have online databases, published literature, some more data sources and everything, and you find all these gene-protein relationships, and you can use then form the stoichiometric matrix of the reaction, and then you can use FBA for finding what is the optimum set of solutions for this particular equation. So set of solutions, when we are talking about a set of solutions, what solution are you trying to find? Mostly the parameters of the differential equation, or mostly the variables in the differential equation. So depending on what you are trying to find, you use FBA in that manner, and you maximize or minimize a particular cost function, and then you do your predictions, right? FBA is used to then predict a steady state flux distribution toward the network given certain underlying physiochemical and thermodynamic constraints. This prediction can be validated with independent experiments, and inconsistencies can lead to model refinement. As we talked about for other systems biology models, it's like you start with a known data, which experimental data, you formulate hypotheses from those data, you do some modeling or analysis from the data, and you propose your hypothesis, and then that hypothesis has to be validated using wet lab techniques, experimental techniques. Now, if that is not validated, you are getting something else, you tune your model in such a way so that you can replicate this particular experimental data that you are finding, right? So, in addition, with the stoichiometric network reconstruction and the FPA, the network can be perturbed in many different ways. So, this is where actually the real application is coming in. So, where should you use FPA? Why do you want to find a set of solutions under different conditions? Mostly this is used is in certain hypothesis creation of like, what happens if I knock this particular metabolite out? What happens if I just delete this particular gene from the genome of this individual? What will happen metabolically? How will the whole metabolic network change if I say remove a particular kinase from the genome, which actually say for example phosphorylates different products in glycolysis? So what will happen if I do that? So these kind of questions can be answered using FPA, and I can perturb the network in different ways, ranging from differences in environmental stimuli to mutations in network structure, which is example is single or double gene knockouts, to assess the properties of the system such as robustness. So what you are trying to say is, if I particularly knock this gene out, will my networks remain the same? Will my metabolic network and the output that I'm getting in terms of the metabolites and the flux, will it remain the same, or will it change a little bit, or will it not change? Some questions like these can be addressed with FPA, right? So this is an example of where FPA is used. So this, I think, if I am not very wrong, this is the glycolysis pathway. Yeah, it starts from glucose 6-phosphate, fructose 6-phosphate. It ends at pyruvate. So yeah, I think this is I don't remember the glycolysis pathway so vividly, but I think this is the glycolytic pathway. Oh yeah, it's written here. Okay. The classical flux balance analysis approach, which actually this is an illustration of a glycolytic pathway as a representative biochemical network. The reactions that convert glucose to pyruvate are labeled from V1 to V12. So all these reactions, glucose to glucose 6-phosphate, this is V1, ATP to ADP. Glucose 6-phosphate to fructose 6-phosphate, this is V2. Fructose 6-phosphate to, I think this is 1,3-fructose bisphosphate, meaning 2 phosphate groups. This is V3, and then you have these V4, V5. All these reactions have been documented. Now, what you do is basically you generate the stoichiometric matrix. So the process of generating a stoichiometric matrix X for a given reaction is demonstrated. Specifically, the conversion of 1 mole of fructose 6-phosphate to 1 mole of fructose 1,6 bisphosphate. So basically this particular reaction, how do you construct the stoichiometric matrix? So here, what are the metabolites that are involved here? Fructose 6-phosphate, which is the starting reactant. I think this the FDP is the diphosphate version, which is the fructose 1,6 bisphosphate. So this is the diphosphate version. So this has +1, fructose 6-phosphate has -1. ATP is getting converted to ADP, so ATP will be -1, ADP will be +1, and a proton is being released, so H plus will be +1. So this is how for each step you can, each reaction you can formulate the stoichiometric matrix, right? Okay. Yeah, so this is this reaction, the conversion of 1 mole of fructose 6-phosphate to 1 mole of fructose 1,6 bisphosphate by the enzyme phosphofructokinase 1, PFK, is represented as a column within S. So this is your S. So each of these reactants will be, or the products or the metabolites will be given as each of the row, and one column will denote a particular reaction, which is in this case is R3. Okay, the rows of S describe the components of the network, while the columns describe the underlying interactions, which are the reactions. The stoichiometric coefficient at the intersection of a row and column quantitatively captures the precise interaction, which inputs delineated and with negative coefficients. For example, if it is like getting used up, you put a negative sign, and if it's getting formed, you put a positive sign. And this is actually, so this is how you make the stoichiometric network matrix. Now it's basically the optimization framework. So this is how you're defining. So max of z is defined as c into v. So this is this particular objective function that you're defining here. And yeah, this is actually a classical depiction of FPA optimization framework, including the objective function. This is your objective function. This is what you're trying to maximize, and the flux bounds that may be applied to the glycolytic network. So this is a flux bound. So you have upper bound, LB is the lower bound, UB is the upper bound, and yeah, C is the vector of your weights, and sorry, yeah, C is a vector of your small c is actually a set of weighting coefficients on the fluxes contained in V. The capital C actually denotes the species concentrations. So basically, concentration of each of these metabolites. M is the set of components, and N is the set of reactions. And this LB is lower bound, UB is upper bound. So you're defining this is all the solution space. You're defining it in that region. You're giving an objective function which is z equal to cv, and you're trying to maximize this objective function, and these are your flux bounds. And if you do that, if you do that, FBA derived flux distribution to the network can be shown. So this, if you model this and you find the fluxes, so in this case it ranges from 0 to 20. So you see the initial fluxes are low. Here the fluxes are very high. So initial fluxes are very low to form a particular element inside the network, or compound inside the network, through which the rest of the reaction will take place, and the flux there is going to be very high. So here also, intuitively also, you can think about the flux balance. So you have a reaction where one part of the reaction has a low flux, one part of the reaction has a high flux. So it kind of balances itself as well. So this is how in general FBA is used to do this kind of analysis of like a network of metabolites, or even like a gene protein reactions. You form the stoichiometric matrix, then you have an objective function that you need to maximize and minimize, and then you also put constraints on different levels for this particular reaction. Now the objective function is actually the most important part of FBA. Depending on how you define the objective function and how you are trying to maximize or minimize that function, your the solutions will be produced in that manner. So you can have good solutions if you are using a good objective function. You will have not so good solutions if you are using not a good objective function. So the FBA model's objective is to alter the distribution of optimal fluxes within a metabolic network, influencing damage accumulation and lifespan, to quantify changes in replicative lifespan and generations time, cell lifespan, using various objective functions in the FBA model was simulated. So this is one example that I'm talking about. This figure I've taken from a paper where they actually showed the different like if you take different types of objective functions, what are the types of results that you can get, and how good or bad can it be. I'll just quickly do a reverse search and see if I can find this particular paper. Not save the picture here, I think. 1 second. Yeah, and this is this paper. Yeah, this actually talks a lot about uh, why objective functions are important and why the choice of objective function is very crucial for understanding the simulations. So this article you can go through if you are interested. Yeah. Okay. Okay. Yeah. So yeah, so talking about the objective function, what are the objectives that you can put in the objective function? It can be maximizing growth rate, minimizing glucose uptake because you want the maximum amount of product in the minimum amount of substrate concentration, so something like that. Maximal minimal and maximal ATP production, or maximal non-growth associated maintenance, something like that. So there are a lot of functions that you can actually optimize depending on your constraint. So if, say for example, as I was saying, production of insulin from E. coli, what you want there is minimizing the substrate concentration, maximizing the product concentration. That is a that is the flux balance that you want to do, right? Flexibility was allowed by permitting violations of optimal values by a factor of eta. So this is for this particular figure, exploring range of values of eta. Glucose uptake rates were solely determined by objectives and constraints, allowing for unconstrained uses. So what the simulation results indicate is that the choice of objective function significantly impacts replicative lifespan and generation times. Cells without maximal growth optimization showed limited growth, while some failing to divide or doing so infrequently with long generation times, emphasizing the importance of maximal growth in replicative aging. So it is this particular example that you have, uh, that you are trying to kind of maximize growth and replicative aging. And if the question or the problem was different, you might be requiring a different type of an objective function. So yeah, so these are all the scenarios actually they have considered. So this is one type of an objective function. This is your first objective, this is your second objective. So you're trying to maximize something. So in the first one, you ended up maximizing this n-gam, which is basically non-growth associated maintenance. And for max growth you had maximum n-gam. For maximum n-gam you had maximum growth, but you could not do for maximum ATP production. So yeah, so determining the objective function becomes a very crucial part of flux balance analysis. How and where you define the objective function will give you the set of solutions that you will or will not probably like, depending on how you have chosen your objective function. Right, okay. Now this is another technique that is also very widely used along with flux balance analysis, which this is known as MUMA, which is minimization of metabolic adjustment. It's basically an algorithm for predicting metabolic flux distributions after a gene knockout. So mostly you'll see MUMA being used in gene knockout situations. So although the assumption of optimality for a wild type organism might be justifiable, the same argument may not be valid for genetically engineered knockouts that were not exposed to long-term evolutionary pressure. That is where this comes in. So basically in flux balance analysis, if you're doing a knockout study and you're trying to find out um, what is the maximum growth or the maximum concentration of a particular product that can be formed, but you assume that it is in an optimal condition even after the gene knockout, which is technically not the case. If in a biological system, if a particular gene has been knocked out, there will be repercussions for that, not in terms of its own particular phenotype that the gene is conferring to, but also in general in the whole network, because that is the whole idea of systems biology, right? So if you knock out one gene, it just does not, like, it will not happen, it does not just form its own product, rest of the things are going on fine, because that gene will be involved in other things as well. So a lot of things will get hampered inside the cell if you just knock out a gene. So the assumption of optimality is not justifiable when it's a knockout. So here the flux balance analysis is not actually a good fit for this kind of modeling. So in the basis of this method lays the assumption that after a knockout, metabolic fluxes undergo minimal changes with respect to the flux configuration of the wild type. MoMA employs a quadratic programming to identify a point in flux space which is closest to the wild type point based on conjecture that the mutant remains initially as close as possible to the wild type optimal in terms of flux values, right? So the idea is that after you knock out, the cell or the system is resilient or robust enough to not change that much in terms of the flux, at least the flux. The other things might change, the products that are getting formed, they might have different kind of characteristics or properties if you do not have an effect of another gene there which was supposed to be there. So that might change, but the overall flux kind of remains the same, or it remains very similar to the initial flux because the system is very resilient, so it will not shift the it will not shift the flux so easily. So that is the idea. So to predict a metabolic phenotype, MoMA determines a flux vector V in the flux space phi of a mutant with the smallest Euclidean distance from a given flux W for the wild type organism. Sorry. So yeah, this is your distance between, this is your older flux, this is your newer flux, this is your distance between the flux which you are trying to minimize. It's as simple as that. So you have older flux, you have newer flux, you take Euclidean distance between them, which is like, you know what a Euclidean distance is, root over of xi minus xj whole square plus yi minus yj whole square, something like that. So this is the Euclidean distance and you are trying to minimize that based on the assumption that your newer flux will not be that dramatically different from your older flux even after a knockout. And minimizing d is actually equivalent to minimizing the square of d, which becomes easier to handle. And constant terms like the wild type flux wi can be left out of the objective function. You can replace wi in terms of vi and other things. So because this is constant, there is no point of keeping it in the objective function. So yeah. Okay, so now we will talk about one good example of like both FBA and MoMA occurring together. So how are the results different and why you should like these are two different techniques. So right, okay, so you have taken a network, you have calculated your stoichiometric matrix, you have calculated your steady state values and everything you have done. You've done the modeling and you have done FBA and MoMA on in two separate occasions, and you're trying to compare the results. This is where it is, right? This is basically so this is all your solution space. So this is basically a schematic 2D depiction of the feasible spaces for the wild type, which is given by phi wt, and for the mutant flux j, and is represented by the green and the superimposed yellow polygons. So this is the feasible space for knockout for this particular mutant, and this is a feasible solution space for your wild type, right? Now the coordinates are two arbitrary representative fluxes, an extremely simplified version of the multi-dimensional flux space. Because remember one thing, here you have only considered two coordinates, because v1 and v2 you have one is a wild type, one is a mutant, so you're comparing both. So if you're comparing say 15 mutants at one time and you're trying to find the solution space, there you have to take the help of linear algebra, because you cannot like physically visualize, because like 15 dimensions and the flux and the solution space between the 15 dimensions, it's like you cannot imagine like that. So visualization is cannot be done. So this is actually a simplified version of that, so that we can understand what is going on here. Now the solution to the FBA problem is the point that maximize the objective function. So this is your basically your sorry, these are your, the red lines are your objective functions, one for MoMA and one for FPA, but like you can just consider the red lines as your objective functions, right? Yeah, the solution of the FPA problem is the point that maximizes the objective function. So this is your FBA space, and this is the point actually, if that is the objective function, this is the point actually that maximizes the objective function for FBA. The solution to the FPA, an optimal FPA prediction can be computed both for wild type and knockout. So this is your wild type condition, this is your objective function, so this is the point. This is your knockout condition, this is your objective function, and this is the point. So A and B, these two are the points, one for knockout, one for wild type, predicted by FPA, right? Now the alternative approach, which is MoMA. The MoMA knockout solution C. Now MoMA gives the knockout solution C here, right? This solution C is calculated through quadratic programming and can be thought of as a projection of the FP optimum into the feasible space of the mutant phi j. Now understand, this is your FPA solution for your wild type, right? If you just project this solution onto this space, so this is one space, the yellow is one space, the green is another space. The FPA solution of your green space, if you project that into your knockout face, which is basically the yellow part, it's actually matching. So what is the story here? The mutant FPA optimum and the corresponding MoMA solution are in general distinct. And so how is the MoMA solution different from your FPA solution? So this is your FPA solution, this is the solution at which this is the wild type solution. The corresponding MoMA solution is actually a projection of this FPA solution onto the particular knockout phase. So these are the 2 points actually that are given by this point is predicted by FPA and this point is predicted by MoMA. Yeah, robustness analysis, if you do for this particular deletions, so you put constraints corresponding to partial gene knockout and you lead to a feasible space that is intermediate between. So if you put more constraint, this is the space that is intermediate between the yellow and the green. So on such intermediate spaces, both the FPA optimum and the MoMA projection can be calculated, and the robustness analysis method is unique to MoMA. One can perform a sampling of the feasible space around the optimal found by the simplex FPA point A, which is this point. So basically you can do a kind of a a vector of the distances between the FPA solutions and the MoMA solutions, something of that order. Okay, the robustness of the MoMA solutions is calculated for defined threshold for sensitivity of the objective function for endpoints obtaining the limited corresponding fluxes to a given fraction of their wild type value. This method is also used for studying possible multiplicity of MoMA solutions in case of alternative FPA optima are present. So these are your alternative FPA optima are given by these 2 points. This is a point, this is a point. So you can calculate the distance between the alternative optima for given by FPA from the MoMA solution. So right, so this is how actually the it's slightly different in terms of, so what you're trying to do, if you recall MoMA, so what you're trying to do is you are trying to minimize the distance. So from this point, this distance is bigger compared to this distance. So that is why the MoMA knockout solution is given here as opposed to here given by FPA. So the mathematics also kind of holds up, right? So the general idea is when you have a knockout, MoMA is a better solution than FPA. But if you have a general problem to be solved in terms of constraints and metabolic networks, FPA is a better solution. Okay, and there's another. I think this is the last part. So on top of optimization techniques like MoMA and FPA, you have this particular algorithm as well, which is known as room regulatory on-off minimization. So basically it integrates regulatory constraints into the prediction. If you remember, So these were the constraints, all these constraints. So we've covered physiochemical constraint, we have used mass balance, we've used topological, not so much topobiological balances, but this can also be used. Environmental constraints as well. Now we are talking about regulatory constraints, right? So yeah, so MoMA minimizes the Euclidean distance between the flux distributions of wild type and the perturbed strains, while Room employs a different norm. So it focuses on minimizing the total number of significant flux changes, emphasizing cell's adaptive cost reduction. So what MoMA does is basically it wants to minimize the distance between the wild type and the knockout. That is what MoMA does. But Room, what it does, it's basically takes into account the total number of significant flux changes. So it will calculate flux after and before, and it will calculate the difference in fluxes between before and after, and it will only take the total number of significant flux changes, and it will try to minimize that. So it's the same idea. The idea is that your new system must not have like a very different flux through the network compared to your wild type system. So this is what it tries to do as well. So yeah, this I'll not go into very details of this, but yeah, so it's another optimization technique. So yeah, so this is another example where basically three of these methods are actually compared. So this is a given flux distribution for the wild type intact network that can be obtained by FP and experimental flux data. The flux through B2, this is the B2 reaction, it represents the growth rate. Now this particular image is basically MoMA's prediction for a knocked out network following the knockout of reaction V6. So this is your V6, so this has been knocked out. So this is the flux that has been predicted by MoMA, and this is the flux that it's been predicted by Room. Okay, it's the same reaction that has been knocked out. This is the V6 reaction that has been knocked out. But there's a slight difference here. Room finds changes in flux only along the short alternative pathway through V5 and V4, preserving the optimal growth rate of the wildlife organism. So what what Room is doing is basically finding a change in flux only along a short alternative pathway. So this is your short alternative. So you're going from here to here. So your alternative pathway is this, right? If I'm right, yeah. So this is your shortest distance if you knock out this particular reaction. So if you should move like this. So what MoMA is predicting is lower flux in these regions in this particular reaction, where Room is predicting a much higher flux and ultimately a much higher flux in the final outcome of the network. Okay, so in all the figures, metabolic branch point B is along the reactions that produced and consumed data are highlighted. Note that in Room's prediction, linearity of the flow is preserved at the branch point B. So this Room is predicting this particular pathway, right? And MoMA is predicting both this and this, because there is a flux here as well. But here this flux is 0. So Room is predicting this pathway, MoMA is predicting both of these pathways. So, but what Room does is it preserves the linearity of flow at the branch point B, as opposed to the MoMA prediction, where it actually branches off the flux at this particular point. Note that also the MoMA solution has an Euclidean distance of 62.8 from the Room solution, and has 3 altered fluxes with respect to A. So if you compare the original with MoMA's prediction compared to Room's prediction, Room only has changed, Room has 3 altered fluxes. On the other hand, MoMA solution has 9 altered fluxes with respect to the original, and but the Room solution has an Euclidean distance of 75 from A, which is lesser than the Euclidean distance predicted by MoMA, because MoMA is trying to minimize the Euclidean distance only. It is not looking at other things. room on the other hand is looking at other things. And this is an example that, like, even if the Euclidean distance is the shortest, that doesn't mean actually that is the best possible solution for the equation, right? Okay, so if you have any questions, you can ask, or we will move on to the assignment questions, meaning like previous year assignment questions. Okay, then I'll put these on screen for a minute or two. You can write your answers in the chat box, or you can, like, since you are only here, so you can unmute yourself and tell the answers as well. Okay, I'll wait for another minute. Just a second, I'll check something quickly. Okay. Okay. Sorry. Yeah, the answer is true. Constraints like physiochemical constraints, like environmental constraints and everything, the only work of the constraint is just to help you reduce the possible space of solutions and, like, help you find a better solution. So that's the idea. So you're reducing the space and you're constraining the solution in a particular space, and you're reducing the area for possible solutions. So yeah, that is the idea. Oh, I think it's the same question.
The second option, 1C, 2B, 3A.
1C, 2B,
Okay,
3A.
no, the third one is B.
Sorry, I did not understand.
Undetermined,
You are saying
once, undetermined would be reactions, undetermined would be no reactions. So, undetermined would be more reactions than metabolites. So, A is
yeah. So when you have underdetermined is basically the number of unknowns are greater than the number of equations, and overdetermined is the number of unknowns is lesser than the number of equations. So that is the idea. So if you have number of metabolites greater than the number of reactions, it's basically it's overdetermined. Yeah, it's overdetermined. I'm saying it right, yeah. And if you have the number of metabolites lesser than the number of reactions, it's underdetermined, because so you have more solutions possible for that particular condition compared to an overdetermined solution, right?
C set of stoichiometric constraints.
Yeah, that's true. It's basically your constraint.
X y dt equal to 0,
Yeah, yeah,
steady state condition.
yeah. This is the steady state condition. Correct. Okay.
I think 1 and 3 are both viable. Not sure.
No. So basically, for different
Oh,
Vmax,
one. Yeah, yeah, right.
you
Yeah,
you you will not get
right.
different
Same
values of.
place.
Yeah. So your R will be unique because it's the maximum flux.
Maximum plus, right, right,
That
right.
will be unique. So yeah, yeah. So yeah, for different Vmax, you can get the same value of R because it's a maximum possible value. So at different Vmax, this particular reaction might be maximized for flux. So it is possible to get. Uh, sorry, sorry, I was stating the opposite thing. So for different Vmax, you'll always get the same value of R because R is the maximum flux for this particular reaction, and it is going to be unique. And uh, like if you take different Vmax, it is possible that you can get different values of R, but if it's uh, if it is satisfying the condition of the maximum flux that is going on, you'll get the same value of R because the R value will be the highest value will be unique. So if you have a different condition where you are getting a Vmax is different, but your reaction is the same, and your that is the particular reaction will have the highest flux, so your R value will be the same and it's going to be unique.
Any of them should be viable.
Sorry.
Any of them should be possible for FP.
Yeah, all of them actually can be undertaking using FPA. So you can max, depending on the constraint thing you're trying to do. Say for example, production or anything, you want to maximize that particular thing, and every of these things can be maximized using FPA. So it's simple. No, you're not writing answers. Okay.
It's
Yeah, it's true. That is the whole idea. The Euclidean distance is basically the distance between the wild type flux and the knockout flux, and you are trying to minimize that. So that is what MoMA is all about. So this is true. I think it's a similar type of a question, but okay, we can answer this.
so 1, 2, and 3.
Yep. Okay, so interactions in a protein network, I don't think you can do it with flux balance analysis. What you can do is you can calculate.
Simply minimizing the parameters.
Yeah, but understanding how it is interacting, it's not for FPA. For that, you need to do some sort of what to say, some sort of normal differential equation based analysis. Ki, why, how is the particular? So, is that in terms of interaction? What you mean by interactions is basically, is this protein enhancing the expression of this gene, or is this protein actually decreasing the expression of this gene, or something like that
Right.
at that level? And
So
FPA
it's
cannot
got
do.
to be prerequisite to FP.
Yeah, yeah. So understanding that actually is a prerequisite.
Going for it.
Oh, 1 second. Okay, 2 and 4. Yeah, that's correct. Yeah. So the third option is actually interesting. Biomass reaction is the only objective function for performing FPA. This is actually true when you are trying to maximize production.
Yes.
So FPA in maximum cases you see is used for maximizing production or increasing biomass or something like that. And in many cases, the biomass reaction is used as the objective function because you're trying to maximize that reaction. So in many cases you'll see this actually, but that is not the only way you can use the objective function. Biomass reaction, other than biomass reaction, you can have other objective functions, but that is also an important objective function in many FP analysis. You'll see.
First one is root 22.
Okay.
First one is root 22.
I
Let
have
me
the
just
answers
start here.
together, so I'll show the answers together only. You can try all of them.
Okay. Yeah, I sent it in the chat. 4.6904, 8, and 3.
Okay. Let's see. Okay. Yeah. So L1, sorry, for L2 norm, it's basically the Euclidean distance. And it's like, I think you give this answer only. For L2 norm is the modulus of the so since it's v1 minus v2, so it's basically x coordinate of v1 minus x coordinate of v2 in a modulus. Similarly for y coordinate, similarly for z coordinate. And if you want to find the infinity norm, it's basically the maximum of these differences, modulus differences. So it's basically the answer is 3 here. I'm not sure. You've given the correct answers anyway. So yeah, that's it from my side. If you have any questions, you can ask, or we'll close the session.
No questions as such. Thank you so much.
Okay, if you don't mind me asking, like, which background are you from? Like, meaning, what is your
I'm currently in biotechnology.
biotechnology BTech?
Yeah, yeah,
Okay, and like you have completed your BTech, and like I'm just asking
no,
because.
I'm still in the third year. I'm doing this for my honors.
Oh, okay, okay. So you're getting credit for this.
Yeah, yeah.
Oh, okay, okay. That's cool. That's cool. Okay, I just wanted to know why you are doing the course. So yeah, that's all.
Right.
Thank you for joining. And if you have any
Thank you.
questions, please actually you can write to me to my email ID if you want.
Okay.
I'll provide it here. Okay.
Thanks so much.
Thank you.
Yeah.
Thank you for joining. Yeah.